anatomy course. There is a fortuitous degree of overlap at this point. So we define the CNS and the PNS. The central nervous system consists of the brain and the cord. The peripheral nervous system is a little bit trickier, but for now we will say that it is everything exterior by exterior, outside the brain and the cord. Last I counted, there were three ways that histologists and neuroanatomists differentiate these two. We won't worry about that now. Let's just make it very simple. The CNS is the brain and the cord, and the PNS is just about everything else. We have another term for a few little special areas, but that's pretty much the case. We have talked about the inherent ambiguities in the term somatic. Just make sure you understand that the term somatic can refer to components of both the sensory and the motor nervous systems. Virtually all motor circuits are going to be somatic. Having said that, I'm going to retract it. Most of the time when you see references to somatic motor, we're talking about the voluntary nervous system. That's what we're talking about. So somatic motor means basically neural signals going to skeletal muscle. I don't think I need to, and we're going to discuss that there are other efferent signals. Somatic sensory, on the other hand, refers primarily to tactile, and we really don't have a word in our English to describe this, but basically sensations that are not special. You know what I mean by the special? Vision, hearing, gustation, olfaction, equilibrium, those are all special senses. Everything else we call a corporeal sense or a somatic sense. And so, for example, again, that would include most tactile sensations. Temperature, vibration, textures, pain, all of these would be somatic sensations. So there's always going to be some ambiguity in the term use of somatic. And then finally, the autonomic nervous system, and I'm not even sure that as I get older, I'm not even sure that I agree with this, is traditionally considered part of the peripheral nervous system. But remember, the autonomic signals, for the most part, originate in the hypothalamus and brainstem. So maybe there's some issues there. And then the two divisions of the autonomic nervous system, as you know, are the sympathetic and parasympathetic. I deliberately avoided the term visceral in the anatomy because I just didn't want to deal with the complexities. But I guess at some point I need to throw the Piper and start addressing it. Yeah.
Yeah.
The information should be the same, and I checked this morning when I brought it up. This is what I saw. Well, it may be the propagation. I don't know. I went to the website. The only thing I can think of, well, even that wouldn't explain it. Are you sure you refreshed your browser? For example.
Yes.
I never opened it.
You've never opened it, and this is what. I think again. Let me just explain this one more time. I can't control this. I have no control over this. When I send the signal to my server, that company has servers all over the world, including one in Miami, and that's why I use this particular server. And my guess is, like we were talking about with divergent neural signals, it takes a while for the signal to propagate to those servers and then come back. My guess is that what we're seeing is the period of propagation. Things like pictures and style sheets, it may take a day. If I change the wording, that's immediate. I can't tell you why that is. It has something to do with the way the coding works in the servers. But in any case, it's not important. And if you can find, this is the challenge I have for you. Find some evidence anywhere in the psychological or learning literature that says you are somehow being deprived or that your learning will suffer. Every piece of literature I have found suggests that the less I give you, the better you do. In fact, if I'm not even here, what does the literature say about most of you are typing into your keyboards there? What does the literature say about the efficacy of typing into your keyboard as a mechanism of learning. It's not the worst thing you can do. It's the least productive, least efficient thing. And they've done this over and over again. Writing, that's why I went to some pain on the tech help page to show you how you can annotate, you can handwrite on the slides. Studies show over and over again that's a lot more effective. And then if I don't give you slides, you're even better off yet. And if I just give you a source and just talk to you, most of the research shows you do better on tests. So all I can tell you is the information is here. It'll be here after, if an hour after class or whenever you check it later today, it's not there, let me know. But I know that it's been up there for several hours. And then finally, everything I'm talking about, we talked about yesterday. There's nothing new here. We've already talked about these main points, so it shouldn't be a real issue. However, the term visceral, yes.
I was just going to clarify something about the autonomic
Yep.
nervous system. It's involuntary, it's a use of motor and
Yeah, so this we talked about yesterday. So the ANS is traditionally classified as part of the efferent peripheral nervous system. So remember our discussion yesterday? We asked, so let's say that your autonomic nervous system right now is regulating your heart rate. What information is it using to regulate your heart rate? Anybody have any thoughts on that? Something as a PA you should think about. What information is it using? Oh, okay. Blood pressure, probably blood gases, probably measure of activity or metabolic rate, all of these things. So that's information coming into the brain that is being used to modulate or regulate the signals from the brainstem that are changing heart rate, changing respiratory rate. So the true answer to your question, independent of what you read in the textbooks, is that the autonomic nervous system is both efferent and afferent. And that gets us back to this word we're talking about right now. So let me talk a little bit more about this role, and you'll see why I really don't like to talk about it. So the term viscous is the noun. It just refers to organs, thoracoabdominal organs. So strictly speaking, when we use the word visceral as a generic adjective, we're just talking about anything other pertaining to, related to the organs of the abdominothoracic cavity. However, here's where things get a little problematic. We've indicated that there are autonomic pathways that go to your GI tract, for example, or go to your heart or your lungs, and their job is to regulate the activity of those tissues. And we've also indicated, in reference to your question, that there are signals coming back from those tissues telling the brain, telling the hypothalamus and brainstem what to do. Those are afferent autonomic signals. Here's the problem. Oftentimes, as we're going to begin seeing, as we begin talking about the head and so on next week, oftentimes the pathways that the ANS uses to bring that autonomic information back to the brain, they follow somatic pathways. What were somatic pathways? Yes. The nerves that are bringing tactile sensations back to your brain regarding touch, temperature, and pain. So let me say that again. Oftentimes, autonomic afferents follow the pathways or follow piggyback. What you usually see is piggyback upon somatic pathways. Everybody understand what I'm saying there? And we call that's one type of visceral afferent. The autonomic return is sometimes called a visceral afferent, but it gets even more complicated. What's going on in your intestine right now? Can somebody give me a complete report? Are you aware of are you any awareness of what's going on in your intestine right now? Sometimes. Give me an example of things that you are aware of, or maybe I wouldn't say that. But what types of things are you aware of from your GI tract?
Shifting.
You said gas. Hopefully you're aware of that before it becomes evident to the rest of us. But, what else? How would you describe these sensations from your GI tract? All right, pressure. Can you say that you have an insect, somebody was saying you've had an insect bites before, if an insect were to land on your intestine, could you specify precisely where that insect is landing? Anybody have a problem with bugs in your belly? Every night I have this issue. Most of us cannot specify. In other words, this sensation is extremely delocalized, just a very generic. Do you feel pain in your GI area? How would you describe the types of pain sensations you have? Often, in many cases, they're not even associated with the part of the GI tract in which they physically originate. We'll talk about that later. It's called referred pain. My point here is that the type of somatic sensations that originate in the viscera are a different sort of animal. They're imprecise. They don't provide us very much information. And the point I'm making is those are also called visceral sensations. They're somatic because you're aware of them, but they don't follow the same pathways. They don't provide the same sensations that your skin does, that your ears do, that your eyes do. Okay. So I hope you see now, when we use the term visceral and visceral afferents, especially with respect to the autonomic nervous system, it's fairly complex and weedy business. It involves anatomical ambiguity and ambiguity with respect to the nature of the sensation. And I think that will become evident as we talk about this more in the regional anatomy. But in any case, I guess for now, we'll just say visceral has two general meanings. It refers to any type of process or event going on in the thoracoabdominal cavity associated with the organs. But when we talk about visceral afferents, what do we mean? What is a visceral afferent? Say you're just as bad off as I am in trying to describe. A visceral afferent, technically, strictly speaking, is an afferent fiber associated with the autonomic nervous system that may or may not communicate tactile or somatic sensations. And that's about as good as I can do for you there. And it gets even worse, as we'll see later. When we actually begin talking about the anatomy. So all of this should look very, very familiar to you. Even if you don't have this slide, you should be able to tell me, you should be able to identify all these structures. For example, what is this in here? What is all that?
Gray matter.
Okay, that would be the gray matter of the cord, and then the white matter would be peripheral to that. We're looking at this. We're looking at a cross-section of the cord here. Can anybody differentiate the sensory side from the motor side? Sensory side is the top or bottom? Top. Sensory is the top. That would be the dorsal side. Of course, you have the vertebra to help you here too. Can you tell me what part of the spinal column? Well, that's true. What region of the spinal column are we looking at? Yeah, cervical. Good. So we can tell just by the shape of the vertebra. We'll worry about that now. You'll learn all that later. So around that transection of the cord, then we're going to have the dorsal and ventral roots. This would be the dorsal. This would be the ventral. And notice we have a bit of an ambiguity here because this pubescence, this enlargement here, is what? It would be that they're intending to tell us that's the DRG here. You can see it's not really on the dorsal root in this illustration, and often anatomically it doesn't appear that way. And so this is going to be our spinal nerve. And then the spinal nerve is going to branch into the ventral and dorsal branches, or anterior and posterior branches. So all of that should be fairly clear. What is this? This thing that looks like what are these? Those, you ever seen this? Bloodworms. Ever seen a bloodworm? I have bugs and bloodworms in my belly. But this thing that looks like a bloodworm on either side of the spinal column, what's that? Okay, good. Those are the sympathetic chain ganglia, sympathetic trunks. Good. And then you can see the vertebrae, the anterior and posterior, and so on. And we talked about this. cervical, thoracic, lumbar, and the sacrococcygeal regions. Again, make sure you know the number of paired spinal nerves in each region. Down here, and I think I have a couple slides on this, make sure you also understand the inherent functional polarity of the spinal cord. Dorsal is central, the dorsal is sensory, ventral is motor, and then the black dot there is what? The black dot is the cell body of the motor neuron. And what is this? The cell body of the sensory neuron for the somatic senses, for senses that bring touch and pain and temperature to the brain and so on. When we're talking about some of the regions of the brain, I'm not going to ask you to identify all the various gyri and sulci here, but you very shortly should be able to identify most of the structures that you see there. And in the anatomy, we'll talk about the curvature of the spinal cord too. We'll talk about how that falls out. We're looking at the cord here. The cord's part of the central nervous system, and you can see again the relationship of the meninges. They talked about the dura mater, the arachnoid, and the pia mater. So in the illustration at the top right here, this sort of outer encasement is the dura mater, and it's being severed here. This glistening sort of coating here, that's the arachnoid. And then as a PA, as a culture, you really don't have to worry about the pia. You're not going to be able to do much about it anyway, and it's irrelevant because it's so thin and delicate. And then we can also see the relationship of the rootlets and the roots to the dura, and it's important to understand that all of these structures extend through the meningeal membranes as they conduct themselves to the various parts of the body. I think you're familiar with the distinction between gray matter and white matter. What do we mean by gray matter in the CNS?
Sensory and motor.
Yeah, mostly cell bodies. Don't get too caught up and don't be too puritanical here, but yeah, it's predominantly neuronal cell bodies. And then the white matter would consist primarily, the reason it's called white is because it consists primarily of myelinated axons. And then out here are the rootlets that we talked about, both dorsal and ventral. You should know the word somatotopic organization. And that's illustrated in the transection of the cord here, and all we mean by somatotopic. Somato means corporeal or body, topic means according to. So by somatotopic organization, the implication is that the neural pathways, indeed virtually the entire CNS, is organized in a manner that reflects the human anatomy. You can see, for example, in the ventral horns here, pathways that are involved in movement of the shoulder here, whereas the pathways that involve movement tend to be more lateral. That somatotopic organization also applies to the brain. This is sometimes called the somatosensory homunculus. You can imagine that little man that you see there spread across the surface of the cortex, standing in front of you. And so what do you notice about the somatosensory homunculus? Now pay attention to a couple things here. What we're looking at is a coronal section of your brain. So this would be the temporal lobe over here, parietal. You can see the longitudinal fissure at the top. This would separate the right and the left hemispheres of the brain. And so when we talk about somatotopic organization, let's say a patient were to have a stroke, and that stroke were to be localized in this region. Where would you anticipate the greatest degree of disability? Yeah, it would probably affect their ability to at least temporarily move their feet, articulate their feet. The other thing to pay attention to as you look at this, and we'll talk more about this later, and notice that your cerebral cortex is primarily concerned about what? It's not too concerned about your feet, your legs, your butt, or your chest. It's concerned mostly about your hands and your head, your hands and your face. Now there is, in addition to that, one other thing. What is the anatomical irregularity, aside from proportion? Is there something here that doesn't comport with human anatomy?
Yeah, the hands are a lot bigger than.
Well, there's a proportion aside, because we've already established that your cerebral cortex, because of the way that we have adapted to the world around us, our hands, our movement of the hands and the mouth is very, very important. But what about just the basic anatomy? Does that make sense to you? Oh, the tongue. Okay. All right, I agree with you there. Most of us prefer that our tongues be inside our mouths. But that's not what I'm doing. There's even something more fundamental.
Like the foot legs are top and the head's bottom. Our head's on top.
Well, I would agree with you. It's a head-top thing, but it's not that one that I'm looking at.
Okay, well then.
So it looks to me the foot's connected to the leg, the thigh, hips there, the trunk. Then what? How? Well, that's true. But in addition to the neck, is the head in the proper orientation? No. So the head is reversed relative to the body. It's not that the body is particular. So, and in fact, I've read in textbooks that this was an error. But no, this is the way your brain is arranged. We don't quite know why. Yeah.
Is there a somatomotor?
Yeah, they're both somatomotor and somatosensory. This would actually be the motor because of an anatomical feature that's present in the somatosensory that's not present in the motor. But I think this is just meant to be an overall archetypical rendering. All right, so we talked about the divisions of the nervous system. Let's talk a little bit about the cells that make up the nervous system. And this is fairly basic. You have this one on the test today, on the little quiz. In general, we're just going to say that there are motor neurons and sensory neurons. We're going to make it very simple at the beginning, just motor and sensory. And as it turns out, these two are going to be sensory, and all of these are motor. All of these are motor. I think we established earlier that a multipolar neuron derives its name from the fact that it has multiple dendrites. So the poles in this case refer to the dendrites. Virtually all these have only one axon, but things get a little complicated over here. We'll come back to that. So the motor axons are fairly straightforward. The motor neurons are fairly straightforward. This is the model that we've been using: a motor neuron with a cell body or soma and sundry various dendrites. Dendrites literally mean just branches. Single axon, intermyelinated axon, the peripheral nervous system terminating in the axon terminals. And of course, we know that the signal is always going to move from body toward the terminals. Same thing here, so it's morphologically a little bit harder to make out. These would all be dendrites up here. Here's our short branching axon. This would be more typical of an interneuron in the CNS. This is a Purkinje cell. It's in the cerebellum. And all of these are dendrites, and it's aggregating, collecting information from a broad variety of cells, and then processing that. This axon is going to take that information that's integrated from all these antennae and send it to what are called deep cerebellar nuclei that are attached to other parts of the brain. All right, so that's multipolar, all motor. These are a little bit more complex. These are our sensory neurons. You can see there are two general types, two general structural types. I just use the word unipolar, but we can talk about why they put the pseudo in front there. And the bipolar. In general, as a gross generalization, the unipolar neuron conducts somatic sensations. The bipolar neuron is more associated with the special senses. You have these in your retina, for example. You have these, the region that you use, the region up there where you collect odors, for example, at the cribriform plate. You have these in the ear. All right, so these bipolar neurons are more associated with special senses, and these are what we've been looking at. For example, this one that you're looking at, where would the cell body be located if it's associated with somatic sensations? Where would that cell body be? Huh? Well, the DRG, the dorsal root ganglion. Everybody see that? And so the reason this is called pseudo-unipolar is because this whole thing is an axon. This whole thing is an axon. It's kind of quibblesome, I think, to call those dendrites. But this whole thing, so in a sensory neuron, remember the information is coming from one direction. It's going to be coming from the periphery, from my fingertips, for example, and then being conducted back to my DRG and eventually into the CNS. So the polarity, the dendrites and the terminals simply indicate the polarity. These would be peripheral, and these would be proximal. So I would expect you to, by the way, this is called bipolar. Bi implying two just means that it has two poles. If you were to differentiate these two, what's the primary? How would you differentiate this one from that?
Myelination.
All right, good. I think that's the way I would do it. Excuse me.
What did you say?
She said I was having trouble with my slide. That in the case of the bipolar, the cell body simply in other words, over here, the cell body is off to the side. In the case of the bipolar, it actually splits. It separates the two poles. So unipolar, one pole. Over here we have two separated by the cell body.
In the pseudo-unipolar neuron, where is the acidoid or
Yeah, I haven't talked much about that, but that's an excellent question. So let's imagine that the dendrites are it's going to be peripheral to proximal. What happens in a sensory neuron is that, it's a little more complicated than this, but that AP begins right here. So that would be effectively the axon pillar. So then it's going to be propagated all the way down. The trouble that I have with John's question is that in some cases, and this is important, in some cases the unipolar neuron is also the sensory structure, it's the receptor. In other cases, it will be attached to a receptor. But in either case, the AP will begin just proximal to those dendrites. So what are the questions about? So what are we looking at? You can see it labeled unipolar here. So what's the function of the cell? Sensory. All right, so this is a sensory cell. So what I want you to be able to do is tell me, identify function with shape. What's the function of this cell? No, that's an efferent. What type of senses would this cell be? Yeah, special. Yeah, so you get the idea. So just make sure you can associate terminology with function. This is sort of a typical representative motor neuron. We can see the multiple dendrites. What you'll notice on these dendrites, though, are what are called dendritic spines. And curiously, at least, oh boy, talk about a complicated topic. What you're looking at in those dendritic spines are the physical substrate of what you know. What we have found is that learning involves a lot of processes. I think I told you I saw an article last week that was very distressing on a number of levels, primarily because it argued that in order for us to learn anything, the cell must break its DNA. Can anybody explain to me, because we haven't talked about it in here, why for you to learn, and of course we end up with the change in synaptic density, spine density, why would it all begin with the breakage of the DNA? And would that entail any risk? Basically what the article was arguing, basically what the study was arguing, is you young whippersnappers can break your DNA willy-nilly, and there are no adverse consequences because you have repair enzymes, you have polymerases that go in there and deal with that. But what happens when you get old? Polymerases don't work as well. And therefore, learning, that's the reason I just stopped learning. I will not learn a darn thing because of the inherent perils involved. Anyone have a theory about what learning involves DNA breaking? What does your DNA do? Basically it codes for RNA and proteins. And so learning involves making new proteins. New proteins, for example, that result in an altered morphology of the dendritic spines. So It's not simply, in other words, your DNA is relatively, well, it's a different issue. Let's imagine what's written in the textbook is true and that your DNA is fixed, all right? So once this is expressed, this isn't going to change very much. In order to change the profile, in order to change the synaptic density and the morphology of the cell, I'm going to have to change the proteins that are produced. You know, I can do all sorts of epigenetic things by turning genes on and off, but that's going to run out of those tricks pretty fast. What learning seems to involve is actually breaking the DNA, altering the enzymes, the RNA polymerases, and so on, and the DNA polymerases that modify the DNA, so that you can actually learn entirely new categories of information, and also, as I've indicated, change the morphology of the neuron. I guess I just was a little upset by the implications of this DNA breakage for old people. Of course, this is a sort of a more typical representation, a more accurate, more realistic representation of a neuron. Now, what would be the function of this neuron? It's a motor neuron. How do you know? Yeah, lots of dendrites. It's multipolar. In fact, can you identify the axon there? I cannot. All right, good. It's good to me. That's fine, probably. But I would be hard pressed looking at that to know if this is the axon or not. It probably is. Yeah.
Would the axon also have almost spike?
Okay, I'm good. That's why your brain is young and mine is not. You see what Luis is saying there? He was noticing that there are more spines here than here. So good on you, Mike. I don't have an answer to that. It's perfectly plausible. Here's a motor neuron at the top, multimodal neuron. We talked about the general organization. Here we can see some presynaptic inputs, capable perhaps of frequency or spatial summation we talked about previously. And you know the unidirectional signal articulated by Bell's law. And of course, in this case, it's going to terminate at a neuromuscular junction, which we'll talk about in more detail later, but we really already talked about it. It works just like the synapse that we've described between two neurons. The only difference being the product will be a contraction of the muscle. In any case, that's a motor neuron. Here's a unipolar neuron. And so once again, notice the polarity of the signal going right on by that cell body or soma located in the DRG. Is this a monosynaptic reflex we're looking at here? Is it a trisynaptic? This is a trisynaptic reflex, correct? Or what is this? The diasynaptic. All right, so to make sure that we have 1, 2 synapses here, 3 neurons. Here's our sensory neuron. Here's our interneuron. Here's our motor neuron. And once again, pay attention to the location of the cell bodies. Again, we've already talked about this in the anatomy. Probably a little bit of an error here we'd like to talk about. So we can see that in this case, the red cell is the motor neuron, the green cell is the sensory neuron. The motor neuron has its nucleus or has its cell body in the ventral root of the cord. And we're looking at a mixed nerve here. The nerve implies that these two are bundled together. Get some type of sensation from the skin. The traditional distinction between the central and the peripheral nervous systems is, I'm afraid, a bit opaque. It is when the oligodendrocytes become Schwann cells. And by that we mean the central nervous system is delimited by the special myelinating cells called oligodendrocytes. We'll look at these in just a moment. The peripheral nervous system is characterized by the myelinating cells we've already talked about, the Schwann cells in our discussion of saltatory conduction and so on. And that usually happens about right here. But by that definition, it's not very precise. I think it's called anybody study that? The Oberlin-Wallenstein law or something like that. Oberliner. I can look it up or you can look it up. But my point here is, how would you classify a unipolar neuron? Is it Central or peripheral? And what is your evidence? What is your...
Peripheral because of the form
We have an argument. What was the reason for peripheral?
with Schwann cells.
Yeah, the cell body is out there in the outside behind that overliner line or whatever it's called because of the, and yet it's traditionally, I hope you can see the inherent ambiguities here. It's traditionally classified, or at least the cell is traditionally classified as central, and then anything distal to that DRG is considered peripheral. This is not a question that I would ask you on a test. I just want you to see the inherent ambiguities. For example, I have down here, the peripheral nervous system does not include cell bodies. What I would have you add, or what I would have you, I guess, add to that or append to that, except for ganglia. Except for ganglia. The ventral cell body is certainly located in the cord. We talked about the inherent problems with somatic. With respect to motor and sensory, I've just have a little table here summarizing. I think most of you appreciate the distinction there, the location of the cell body, the direction, the orthogonality of the signal, which way it's moving in relation to the cell body. You know where a sensory neuron and a motor neuron terminates. Afferent neurons, with the exception of visceral afferents, tend to provide sensations. By the way, can anybody define a sensation other than except a kid's name is always getting in trouble?
Justin Bieber.
Yeah, he's a sensation, I don't know. I'm saying for Justin Bieber, that is a sensation. And as long as these terms we use all the time, they're not so easy to define. A sensation, okay, what's the difference between a sensation and a perception?
Perception's not real.
You say perception isn't real?
Like a perception is the difference between every one of us, but a sensation, heat, sensation.
Well, I would say that even the sensations could be different. For example, our color sensations, right? We've had the battle over the wedding gown. Or what is, I'm cold all the time. I have to wear, and probably for a lot of you it's hot in here. Yeah.
Sensations are again internally and perceptions of the external environment.
Well, that brings up a good question. Can you have a psychogenic sensation? Can you have a sensation that does not have a physical or stimulus substrate? Yeah. So that might
Would that be
have.
like emotion? Is that what you're talking about?
No, but I wouldn't quibble with you. I mean, I get all emotional with Justin Bieber. No, I was thinking about something like phantom pain, for example.
Okay,
Yeah. The traditional distinction is that you are conscious, you are aware of the perception. Are you aware right now, before I even say anything? And how can I say what I'm going to say without saying it? But are you aware of the clothing on your shoulders? Are you aware of the pressure on your butt right now because you're putting all that weight on your legs? You know, I said something. Are you aware of the fact that you need to go to the powder room right now because of the bladder distension that you have? So you have all kinds of sensations, but you need not be aware of them. But it's very hard to articulate. It's kind of like pain, you know, what constitutes pain. But in any case, perceptions are sensations that are brought to consciousness. And let's not even talk about consciousness. Well, there's a couple more terms I don't know. Yeah, I think this is addressed elsewhere, but I do want you to pay attention to this terminology down here. So, in your line of work, you're often going to be dealing with motor pathway diseases, motor diseases, and sensory paradoxes. And we're going to talk about the primary sensory pathways later. You're going to have to learn the spinothalamic tract and so on. But for now, let's just think about it with respect to what we've talked about. When you move your fingers, use the lumbricals or whatever, where is the neuron located that's responsible for actuating or contracting those muscles? Where is the neuron located? All right, in the cord, in the ventral horn of the cord. All right, we call that the primary motor neuron. It's called it goes by a variety of names, you really need to know them all. Like the sympathetic chain ganglion. It's called the primary neuron. It's called the LMN. What does LMN stand for? Lower motor neuron. Lower motor neuron. You'll study all kinds of lower motor neuron diseases. In fact, in Australia, they call Lou Gehrig's disease lower motor neuron disease. And it's also called the alpha motor neuron. The alpha motor neuron, yeah.
What was that first one? Primary motor neuron?
Primary. The primary is more descriptive and literal. These other terms are used physiologically and clinically. I just want you to be aware of it. All right, so, and why is that? Because it's also the final common pathway. It's the executor of your brain's will, if you will. Now, by contrast, can anybody tell me where the primary sensory neuron is? Where's your primary sensory neuron? What is it? All right. Trenton says it's the one that conducts the signal back to the spinal cord. So it's really the first one, right? So it's just the opposite of the motor. Motor neuron, it's the last. In the sensory circuit, it's the first. But we call both of those primary. Is that clear what I see there? Okay. The title of this slide is Pseudounipolar Neurons and a DRG. And these would be the neurons here, neuronal cell bodies. You see satellite cells. These are a type of glial cell we'll talk about a little later. But my question for you as you look at this, are there synapses in a DRG? Are there synapses in a DRG?
Start with the DRG.
My own opinion, to the extent that I'm able to detect it, is that there are no synapses in a DRG. Everybody agree with that? Why are there no synapses in a DRG? What does a sensory ganglion consist of? Yeah. Exactly right. So the DRG is just an aggregation of cell bodies, sensory cell bodies. So in some respects, it's not even a true ganglion. What type of neuron is this? How would you classify it? Multipolar, what's its function? Motor. All right, good. So you shouldn't have any trouble making these morphological assessments at this point. This is a tricky one. We're looking down the barrel of a neural structure. When I say down the barrel, we're looking at a transection. I guess that's obvious. So we have a neural structure and we've cut it. Anybody have an idea what we're looking at here?
Okay.
What? Oh, vesicle. So when you say vesicle, what types of things would be in this vesicle? All right, so good. All right, excellent. You're exactly right there. These little white, these little circles would be axons. The black is the fat that's stained with some type of metal stain, because that's all that you can really use in most cases to show up. So this would be a nerve or vesicle. And we're looking at axons. And my point here is I just want you to understand that a nerve is a bundle of axons.
You said that black is fat, not
Or myelin, yeah.
mild?
Yes, myelin is a specialized sphincter lipid. But the reason I say fat is because when we talked about this, we talked about the unilocular fat. When you try to stain it, you try to fix it and stain it, you use an organic solvent, all the fat leaves, and all you're left with cell membranes. So it's typically what they'll do is use some type of metal to displace the fat, and it's very dark and opaque for that reason. So again, just make sure you understand the definition of the nerve, or whatever, whichever term we talked about, the endoneurium, perineurium, and epineurium earlier. That it's a bundle. It's like the wiring harness in your car, right? It's a bunch of axons or conductors taped together, joined together. Technically, there are no neuronal cell bodies in a nerve. And we usually reserve the term nerve for the peripheral nervous system. This is an axon terminal. This is an alpha motor neuron terminating. You can see the axon terminal here. This big thing up here, you can sort of see the striations. That's a motor. A skeletal muscle cell. And this is one here too. You can hear it's densely stained in this illustration. But this is the neuromuscular junction. You can see individual motor endplates. Here's our motor axon. Going down to this point of excitation, the point of interaction between the motor nerve and the muscle cell. And you can see the definition that I want you to be familiar with here. It's called a motor unit. A motor unit is one motor neuron and all of the skeletal muscle fibers innervated by that motor neuron. Motor unit. I don't know where to put this slide because it's inevitably confusing for all of us. But we're going to shift our attention now, and we're not talking about neurotransmission. So axonal transport has nothing to do with neurotransmission. What we're talking about here in axonal transport is the fact that these neurons are typically quite long. We've indicated how long they are. They extend, for example, from, in some cases, the brain out to your finger, from the spinal cord out to your toe, and so on. So they're quite long, consisting of these very long axons. And that presents special issues for the metabolism of that cell. In particular, the cell is constantly generating, the cell body, constantly generating protein products, for example, various other products that are necessary at the axon terminal. All right. There are two fundamental mechanisms, at least in our discussion, two fundamental mechanisms that enable these products of the endomembrane system, such as you see here from the Golgi body and so on, to make their way all the way down that axon to the axon terminal. These could be raw materials for neurotransmitter synthesis or transporter replenishment or whatever. The two mechanisms of axonal transport sort of follow the description we used before, fast and slow. Fast and slow. Slow is basically, it's the material is just floating in the current. It's kind of like throwing a paper cup into the Coosawhatchee River and watching which way it goes. It's going to vary with the tide and various other processes. And pay attention to the rate of slow axonal transport. Notice the unit of time here. We're talking about typically 1 millimeter per day. 1 millimeter per day. Now, we have a couple of animations. I think we'll just wait off on these. But the fast works by an entirely different mechanism. We'll look at a couple of videos about how it works next time. But for now, notice the difference in time. It's about a half meter per day. A half meter per day, not millimeter. So that's much faster, much more rapid. We'll talk about the mechanism by which that occurs next week. All right, that's all.